The termination step of prokaryotic translation. This step actually leads to finally what is called as ribosome recycling. So ensuring that the entire translation complex is dismantled so that translation can initiate again. So let us look at the learning outcomes of this session. It is interesting to note that there is no tRNA that is specific for stop codons, and hence, when the ribosome has a stop, arrives at the stop codon and is present at the A site, no tRNA comes into the A site. There are two classes of release factors comprising RF1, that is release factor 1, release factor 2, and release factor 3 that play a significant role in the events of termination of translation. All these class 1 release factors are responsible for release of the polypeptide from the ribosome complex. In fact, it is at this stage that actually termination has stopped because the polypeptide that was synthesized by the ribosome has been released. Another factor, ribosome recycling factor, is responsible along with elongation factor G for disassembly of the entire translation machinery, bringing translation to a final end. What are the key regulatory components of termination? So there are what is called as release factors, and these release factors are able to recognize the stop codons and also hydrolyze the polypeptide from the tRNA. So there are two classes as mentioned, class 1 and class 2. Class 2 in turn has release factor 1 and release factor 2. Release factor 1 can recognize UAG, stop codon, while release factor 2 can recognize UGA, stop codon. And UAA, the third stop codon, is recognized by both RF1 and RF2. Now, both RF1 and RF2 can occupy the A site, and this is so because they are mimicking a tRNA, another example of molecular mimicry. These RF1 and RF2 along with the RF3 are able to detach the polypeptide from the ribosome. And also what is observed is this RF3 is responsible for dissociating the RF1 or the RF2, either of which is present in the A site, that is released thanks to RF3. So now let us look at RF1 and RF2 and look at its structure and function. Now, we already have observed that or understood that RF1 and RF2 are mimicking tRNA. So let us look at how. So you know that a tRNA can bind to the mRNA by the anticodon-codon interaction. Now, interestingly, what has been observed with respect to RF1 and RF2 is that the RF1 or the RF2 is also able to interact with the mRNA, but the codon here is the stop codon. So both RF1 and RF2 can interact with the stop codons either by interacting directly with the stop codon or by interacting with 16S rRNA that is present in the decoding center in the 30S. Now, because of this positioning of the RF1 and the RF2 at the at this domain of the RF1, which is positioned close to the decoding center of the ribosome, is present three amino acids which are conserved. In case of RF1, it is basically serine, proline, and phenylalanine. So this is considered to be the putative tripeptide anticodon. Okay, and at the other end, okay, is present the motif which carries a conserved GGQ, that is glycine glycine glutamine. And this GGQ or this motif, especially the GGQ, is primarily positioned close to the peptidotransferase center. And you can see how, like the 3' end of the tRNA is positioned on the peptidyl transferase center, so is this motif containing the conserved GGQ positioned close to the peptidyl transferase center. So if you can effectively see in the tRNA, the anticodon arm is bound to or is present near the decoding center, while the 3' end which carries the amino acid is present near the peptidyl transferase center. So this is present in the 50S and this is present in the 30S. So is the RF1 and RF2 with this domain present at the 30S near the decoding center and this domain present at the 50S near the peptidyl transferase center. So that is the reason why you can see how the RF1 and the RF2 is mimicking the amino-acylated tRNA. So this, as I mentioned, is what is called as the putative peptide anticodon. And this GGQ, which is conserved, plays a role in breaking the ester bond that is there with the tRNA carrying the polypeptide. And basically, this GGQ is responsible or helps in the release of the polypeptide. So this has a very important role to play. What is also observed is that the GGQ, which is present close to the peptidyl transferase center, and helps in the hydrolysis of the acyl bond that is there between the polypeptide and the tRNA, requires water. So it has been observed that if the Q is changed in the GGQ, then this specificity of needing water to carry out hydrolysis is affected. So therefore the Q of the GGQ is able to specifically interact with water and enable the hydrolytic cleavage of the acyl bond between the tRNA and the growing polypeptide. So let us look at how RF1 and RF2 interact with the ribosome. So this is where we were, where you have the translocation of the ribosome leading to a condition where at the A site is present UAG, which is a stop codon. At the P site is present the tRNA carrying the polypeptide that has been synthesized, and at the E site is present the tRNA that has shifted from the P site to the E site. Now, what effectively happens is that if this codon was not a stop codon, you would have had a amino acyl tRNA coming and binding to the A site and then accommodating itself to ensure that the amino acid that it is carrying is close to the peptidyl transferase center for another peptide bond to be formed. But as we understand, the stop codons do not have a corresponding tRNA, and therefore you will not have any tRNA coming in. Rather, what will come in is the RF1 or the RF2. If it is UAG, what comes in is the RF1. If it is UGA, what comes in is RF2. And if it is UAA, either RF1 or RF2 can come. Like you can see, it positions itself in such a way that this domain is clear to the decoding center and this domain is in proximity to the peptidyl transferase center. Now, what was earlier understood is that when you have the SPF interacting with the UAG, this interaction is important for uh, uh, again, and please do remember that this interaction is a protein RNA interaction. Whether SPF interacts with the mRNA or it interacts with the 16S rRNA, it is going to be a protein RNA interaction, and this protein RNA interaction is specific. So when you have this protein RNA interaction happening, automatically what happens is that this domain that carries the GGQ is very close now to the peptidyl transferase center. So this binding is important for positioning of the GGQ near the peptidyl transferase center. And this interaction that it can make with the tRNA at the P site, its 3' end is close to the GGQ moiety, and this is where the acyl bond is between the polypeptide and the tRNA. So you can see very strategically the GGQ is placed very close to the bond that has to be cleaved. Now, using water, this acyl bond can be hydrolyzed and the polypeptide can be released. So this primarily what was proposed and also reported was happening. But a lot of studies thereafter have basically suggested that there is actually a different mechanism, not a different mechanism, but a mechanism that is more clear now. Now, before we go into that mechanism, let us look at the structure of the RF1 again. So, this structure, as you can see, shows that the GGQ conserved 3 amino acid sequence is very close to the SPF. But when you look at it in the ribosome, you know that SPF and the GGQ are at opposite ends. So therefore, what has been suggested is that when the RF1 or the RF2 are not bound to the ribosome, then this is the conformation in which it exists. But when it binds to the ribosome, then its conformation changes such that the SPF is now going to be interacting with the mRNA or the 16S rRNA, and the GGQ is going to be close to the P site tRNA at its 3' end and also close to the peptidal transferase center. So this also gives an indication of one other factor, and that is that the RF1 or the RF2 will not come and interact or not come and occupy the A site unless and until there is a stop codon. Because if there is a stop codon and when it comes and binds to the ribosome, the SPF is able to bind to the stop codon, it will form a conformation like that which is more stable. Otherwise, it would go back to this conformation and thereby not bind to the A site at all. So if there is AAA over here and if RF1 or RF2 come in, because they are not able to make an interaction with the AAA, they will not change conformation as much and therefore they will move out. So therefore, that has been extremely important that these RF1 and RF2 are able to occupy the A site because they are able to interact with the stop codon at the decoding center. So this is something that has been observed. Now, another thing that has been observed in relation to RF1 and RF2 is that they also act as what is called as post-translation quality control uh, molecules. Now, what does that mean? Say, for example, even after the ribosome has tried to ensure that an incorrect amino acyl tRNA has not entered the A site, supposingly, for example, here there is an ACC, okay, and the anticodon should ideally have UGG. But say, for example, you have UGA and an A and a C cannot interact, then there is a mismatch over here, be it in one base pair. So therefore, what has been observed is such mismatches have been recognized by the RF1 and the RF2, and before translation ends, it basically, or before the peptide bond forms, it is trying to basically look at whether there is a fault or not. But exactly how does it work, we are still not very clear, or studies are going on with respect to this aspect. Now, as I mentioned earlier, the mechanism, molecular mechanistic that is now observed to be happening because of various state studies conducted is what is going to be proposed now. So we already have the 70S with the tRNA at the P site carrying the polypeptide, and at the A site is positioned the stop codon. So effectively, an RF1 or an RF2 is going to come and bind to the A site, interacting with the stop codon at one end and interacting with the peptidal transferase center at the other. Now, what comes in next is another RF release factor, and this belongs to the class 2 release factor. So, this is what is called as RF3. RF3 are GTPase, inherent GTPase molecules, that is, they are able to act as an enzyme. So, the GTPase activity of the RF3 plays an important role in this termination step. So, the GTPase activity is responsible for converting or hydrolyzing its GTP into GDP. Another point that has been reported is that this RF3 has equal affinity for GTP and GDP. So, whether it is present as RF3 GDP or whether it is present as RF3 GTP, it can come and bind to the ribosome at the uh, factor binding center. This is something that has been stated. So, let's consider that RF3 GTP has come and bound to the factor binding center. Now, what is interesting to note over here is that generally all G proteins that are associated with translation, when they come and bind to the factor binding center, due to the change in conformation that happens when they interact with the factor binding center, the GTPase activity is initiated. But in case of RF3 GTP, that is not what is happening. So its GTPase or its GTP conversion into GDP is not based on ribosome. So it is ribosome independent GTPase activity. Okay, but when you have this RF3 GTP coming and binding to the ribosome, two things can happen immediately. One is that the polypeptide is released, and second is that you will have the RF1 or the RF2, whichever was associated with the A, getting moved out. Now the question is, why does this happen? That is because the RF3, when it binds to the factor binding center of the ribosome, it brings about a major conformational change in the ribosome. In fact, what has been stated is that there is a rotation that happens in this ribosome, and that rotation is actually uh, creating an environment that leads to the polypeptide being released by the ribosome. So it is basically the 23S rRNA that is present in the peptidal transferase center and the RF1, which was present over here, that interacted closely with this tRNA to have the acyl bond broken. Now, once the RF1, and of course, because there is a conformational change in the ribosome, the RF1 is immediately, or the RF2 is immediately displaced. This would not have happened if you don't have the RF3 GTP coming and binding. Now, with the RF1 having gone, the polypeptide gone, there is going to be another slight conformational change. And because of that conformational change, you have the RF3 getting released. But before that, let's consider that if you have RF3 present in the GDP form, because what we have mentioned is that RF3 can exist both as RF3 GDP or RF3 GTP. And when it is present as RF3 GDP, even RF3 GDP can go and bind to the ribosome. Okay. But as RF3 GDP, it is not able to dissociate the RF1 or the polypeptide. It has to be converted to RF3 GTP. So that conversion into RF3 GTP is carried out by guanidine exchange factors. So there are several guanidine exchange factors present. So they just exchange GDP for GTP. And so this RF3 becomes GTP. So RF3 GTP then is able to release the polypeptide and also the RF1 or the RF2, and then it itself releases from the ribosome. So therefore, eventually what you are left with is what is called as the post-translation complex. Okay. So this is what is now present. And effectively in the last step itself, termination was over because the polypeptide was released. But now what is going to happen is something that is important for translation, and that is that this entire termination complex has to be dismantled. Why does it have to be dismantled? Because if 50S remains associated with 30S, then another cycle of translation will not happen. When one needs to initiate translation, one only needs the 30S. So the 50S has to get dissociated, which means what we are looking at is that this entire complex containing the tRNA associated at the E site and the P site to the mRNA, which in turn is associated to the 30S and the 50S. So this entire thing has to get dismantled. And this is carried out by three molecules, in fact. One is what is called as the ribosome recycling factor. One, of course, we already know about is elongation factor G. And the third molecule is IF3. So let us just look at the ribosome recycling factor. So the ribosome recycling factor, as you can see, its structure is again very similar to tRNA. So an inverted L. So you can again see molecular mimicry here. You have the RRF mimicking a tRNA, and also the helices that are present in the RRF are able to make very specific interactions with the ribosomal RNA in the ribosome, especially with the large subunit ribosomal RNA close to the A site. So therefore, understandably, it is this ribosome recycling factor that can enter the A site due to the conformational changes that have already occurred in the ribosome. It is able to have an affinity to bind to the ribosome recycling factor. And when the ribosome recycling factor binds to the A site, the ribosome recycling factor itself can bring in more changes in conformation of the ribosome. So you can look at this that with every step there is some change in the conformation of the ribosome, and that change in the conformation of the ribosome has played an important role. So you can see that one after the other, as molecules keep binding to the ribosome, they're effectively changing conformations, and those changes in conformations have led to one event going to the other, to the next, and so on. So now let's look at what happens. So you have the ribosome recycling factor coming and occupying the A site, thanks to the fact that it is associating with the ribosomal RNA present in the ribosomes, and because it interacts with the ribosomal RNA, it brings in a change in conformation. Now, because it is mimicking a tRNA, what comes in is a elongation factor G. All of us know that elongation factor G is again having an inherent GTPase activity. When it is bound to GTP, it has a conformation which enables it to bind to the factor binding center. When the when the EFG GTP binds to the factor binding center, its GTPase gets active and it converts the GTP into GDP. So therefore, when you have a GDP associated with EFG, you can see immediately there is a change in the conformation, and in this conformation, the EFG GDP is actually mimicking an EFTU GDP tRNA. So therefore, when it tries to occupy the A site, it is going to do exactly what it did to the amino acyl tRNA that is present in the A site during translocation. So this EFG GDP tries to push the ribosomal recycling factor to the P site. So this is what is going to happen. Now, when you have this pushed to the ribosome recycling factor, again the EFG GDP's affinity to the ribosome decreases, and also what happens is, because this binds to the P site, you have both the tRNA being released from the E site and the P site. Now you can understand that here the P site tRNA does not go to the E site and then gets exited. Here you will find that both the tRNA which were originally present at the E site and the P site leave immediately. That's because there is a conformational change in the ribosome that is not having affinity for the tRNA to bind. So the pockets are affected, and because the pockets are affected, the tRNA go away. So with that, you can see that the 50S rRNA is dismantled from the 30S. You have all the factors that were associated with the decoding center and the peptidyl transferase center also leaving, and then you have the mRNA also leaving the 30S. Now what happens is that the IF3 comes and binds to the E site of the 30S. This is important. Why is this important? Because if an IF3 is bound to the E site at the 30S, then it will not allow the 50S to bind to the 30S. And this is exactly what is needed for ribosome recycling, because this 30S with the IF3 is again becoming ready for the next cycle of translation. So this entire process of RRF, EFG and the IF3 coming in and associating themselves with the with the translation complex leads to what is called as the ribosome recycling. So let us make the conclusions. The RF1 and the RF2 of the class 1 release factors are able to interact with the ribosome, and they come into a conformation such that its tripeptide anticodon, that is a putative tripeptide anticodon, is near the decoding center of the ribosome, and the GGQ, which is again conserved, are strategically positioned at the peptidyl transferase center of the large subunit. The class 2 release factor, which is a G protein, that is the RF3, which is a G protein, ensures the release of the polypeptide and also the release of the class 1 release factors. To the post-termination complex will come and bind the RRF that mimics again a tRNA. The EFG, which again mimics EF2 GTP tRNA, and both of these then will bind to the factor binding center and push out the RRF, and eventually changing the conformation of the ribosomes in such a way that everything gets disassembled. The IF3 then comes and binds to the E site of the 30S subunit, ensuring that the ribosome recycling happens. Thus, termination step is a cascade of events that leads to the release of the polypeptide and disassembly of the entire translation complex to ensure that ribosome recycles to initiate translation all over again. Thank you.